on precision medicine in IBD, titled What's Right for Me? Precision Medicine in IBD. My name is Antonella Cardone, and I will moderate the discussion today. So this workshop is organized by IFCCA, the International Federation of Crohn and Colitis Associations, within the METHYLOMIC project. It is the second annual workshop organized within this framework. Last year, we explored the role of artificial intelligence in IBD. And this year, we are taking a step forward by focusing on how precision medicine can transform the way we understand, manage, and personalize care for people living with IBD. I guess we now really need the housekeeping rules, as we need to make sure that everyone is muted. Please mute yourself. Because otherwise there might be background noise that can disturb the conversation. You must remember that this webinar is recorded. So if you do not agree, because you already provided your consent, but if you do not agree, this is the moment to leave. As it is recorded, we ask the participants to keep their camera off, so to keep some order on the screen. There will be a Q&A session, and you can raise your hand if you want to ask a question, or please use the chat to put comment or questions there, and you can actually start familiarizing with the chat as we go along. Next slide. So this is the program for today. I will set the scene, and then we will have some presentations to get into the content of precision medicine in IBD. And then we will have a short break, and then after the break we will have an interactive discussion. We will have a panel discussion with the speakers and with the audience, and then the conclusion. So I will now introduce you to the content of today as some background on the METHYLOMIC project might be relevant, so to get everybody on the same page. So the METHYLOMIC project is funded by the European Commission through the Horizon program. It began in 2023 and will run through December 2026, so for a total of 48 months. The project involves 18 partners from 9 countries, and the consortium includes experts in clinical research, epigenetics, DNA diagnostics, patient organizations, and industry innovation. The project is coordinated by the Academic Medical Center at the University of Amsterdam under the leadership of Professor 𝗪𝗲𝗃. And the overall aim of the project is to improve treatment for immune-mediated diseases, such as Crohn's diseases, rheumatoid arthritis, and psoriasis. And although biological therapies such as adalimumab, vedolizumab, and ustekinumab are commonly used, they do not work for everyone. In fact, less than 40% of patients respond to any given therapy, making it extremely difficult to predict which treatment will be effective for each individual. So this results in long-term disease complications and increased healthcare costs. And the METHYLOMIC project aims to identify predictive biomarkers, particularly epigenetic markers, such as DNA methylation, which may help forecast a patient's response to a specific therapy. By using advanced machine learning, treatment responses to biological therapies can potentially be predicted with up to 93% accuracy. And the project is committed to bringing this personalized medicine approach into clinical practice by developing a rapid test capable of predicting treatment outcomes for patients with chronic diseases. And validation studies are being carried out with experts and patient organizations across Europe. And the project will also test this approach in a clinical trial. The ultimate goal is to achieve regulatory approval and develop the first personalized epigenetic test for Crohn's diseases. So now we get into the content of the project, of this webinar. So I give the floor to Dr. Virginia Solitano, who from the San Raffaele Hospital in Milan, and she will present on lessons and failures in IBD, precision medicine, and the new Omicron study. So Virginia, the floor is yours, if you can.
Thank you. Thank you for the introduction. And just
so while you take, while you get
okay.
the slides on, I will mention that you will be speaking about what is precision medicine, how clinicians currently choose the treatments, and an overview of the past attempts to personalize IBD care. So
okay.
the word, Virginia.
Thank you. Can you see the screen?
Yes, we can.
Okay. So thank you again for having me. And today I will talk about what we have learned from precision medicine in IBD and what has not worked so far, and how the Omicron study, that's part of the METHYLOMIC, is trying to change the way we choose treatments. Before we get into the study, it's important to reflect on why precision matters in the first place. So IBD is not one single illness, and even if two patients share the same diagnosis, their biology, symptoms, and response to treatment can be very different. And that's why precision medicine tries to tailor therapy to each individual patient. In this slide, around the circle, you see a wide spectrum of manifestations, from endoscopic images to radiology, involvement of other organs, extraintestinal features. So all these images remind us that IBD is not a single disease, but a collection of diverse biological and clinical expression. However, as clinicians, we know that our current approach does not always take into account this diversity. Despite our progress, we base our decision in clinical practice on a trial and error approach. This means that when we pick a drug, when we select a drug, we base our decision on the available scientific evidence, but actually we try and we wait for some months to see and to evaluate the effectiveness of the given drug. So this is not exactly aligned with the idea of precision medicine. That's to provide patient with the right therapy at the right time. We now have many treatments, and this is great, but this also means more uncertainty. Which drug is right for which patients. Many more are being tested, and precision medicine should help us make the right choice before treatment begins. So from experience so far, we have learned that IBD is not one disease, but it includes many biological subtypes. We also know from evidence that clinical symptoms do not always reflect objective inflammation. And from several trials and from observational studies as well, we learned that early effective therapy improves outcomes in the long run. So this means that the sooner is the better, especially for Crohn's disease. However, we still rely on a reactive medicine approach and not on a predictive approach that is what precision medicine is actually asking for. As mentioned already, 40 to 50% of patients lose response to biologics over time at 1 year. This is the rate at 1 year. And we lack reliable tools to predict which therapy will work. We have biomarkers in clinical therapy, but they are not yet fully precise. We need better biomarkers, tools able to combine multiple sources of information, including genetics, immune signature, clinical data, prior medical history of patients in order to predict flare, in order to predict treatment response. So many biomarkers don't work now in real life because, as I said, patients are different, and also because a signal that is associated with response doesn't necessarily predict response. We also lack for standardized protocols and shared data sets that scientists can work on together. Another reason why we lack biomarkers is that a single biomarker cannot capture a complex disease like IBD, and we need a multidimensional signature that reflect real biological complexity. And something new is coming up. Advanced computing and artificial intelligence can help us to read these complex patterns, and this may transform multiple signals into something that is useful for clinicians. And this brings us to the Omicron study. This is part of the METHYLOMIC, and it's a new international effort combining omics data with clinical outcomes in Crohn's disease. The study is testing an artificial intelligence model to predict therapy response to two common therapies, vedolizumab and ustekinumab. The idea is simple, and it is to predict therapy effectiveness with high confidence through an artificial intelligence model. This is the study design. Almost 400 patients will be included. And before the treatment starts, some blood samples will be taken. And the AI reads the patient DNA methylation profile that is like a biological fingerprint. And this is predicting which therapy is most likely to work. The other arm, in pink, is gonna receive the drug based as per clinical practice based on the physician assessment. Methylation may sound as a difficult term, but this is a chemical modification of our DNA. It happens in everyone, and this mark does not change the gene, but is changes how genes behave without changing the structure itself. So it's the addition of these chemical groups changing the expression of some genes. So the AI learns to read methylation patterns in the Omicron study, and it's gonna distinguish between patients who will respond to those who do not. So this is hopefully turning biology into prediction. So if we can predict the best therapy before starting the treatment, we can definitely avoid delay and reduce the burden. So the ultimate question this study is gonna answer is whether this AI tool looking at DNA methylation is able to predict the most effective treatment in patients with Crohn's disease. So precision medicine is not about more choices. It's indeed the right choice for each patient. And in this study, AI will help choose the right treatment, not replacing doctors, but supporting a personalized care, ultimately improving patient care. So with this, I will hand it to Andrew, who is gonna talk about where to find biomarkers for precision medicine.
Okay. Should I then continue on with my presentation? I will, apologies, I'll just start sharing my slides then.
Antonella, you are muted, sorry.
So very briefly, thanks. So very briefly, Professor Andrew Lee Im comes from the University Medicine in Amsterdam.
Okay. Yeah, thank you. Thank you for having me here as well. I think Virginia did a very great job at explaining what the concept is of personalized medicine. And she gave a very good example of how we in the Omicron trial / METHYLOMIC project try and utilize epigenome to predict patients. But let's see whether that is the only modality that can be used to understand IBD. Because I mean, if we take a step back, if we look at precision medicine, like Virginia said, we have, I would call it in a very, well, let's say, flat way, conventional medicine, a uniform treatment regimen based on treating clinicians' experience, which is just in a very flatly put a one size fits all. Whereas precision medicine would be tailoring the treatment to the individual based on certain features or characteristics. If you were to look in the Oxford English Dictionary, they describe it as the medical care designed to optimize efficiency or therapeutic benefit for particular groups of patients, especially those using genetic or molecular profiling. And this, I feel, is the key behind the precision medicine. However, when we try and go for this so-called molecular subclassification of patients, I have to say that it is easier said than done. But let's start with the various modalities that we can look at. The first one being the most obvious, genetics. I mean, genetics, very simply put, is the heritable material in the form of 𝗪𝗪nucleic acid that contains a blueprint of the data. I think that most of you, the audience, know what DNA is, but suffice to say, it is where all the information comes from. The information flows outwards. And back at the turn of the century, it was thought that if we were to map the human genome, we would have the answer to all. We would understand human evolution, the causation of disease, and the interplay between an environment and certain human conditions. They were so-called thought to be the golden bullet. But as with any hype, that did not seem to hold up in the long run. We found that disease is a lot more complex than simply looking at the genetics. Nonetheless, we should not forget that the genetic variants that have been identified in relation to IBD have paved the way for the development of drugs such as the 𝗪𝗪𝗏 pathway, leading to the development of, for instance, 𝗪𝗪𝗏𝗏, but also the more novel variants found in the TL1A pathway, which is, well, one could argue, the new kid on the block in terms of treatment. But the thing about genetics is it's rather difficult to change it. So genetics can be used in establishing whether a patient will have IBD or not, or whether a patient will respond or not. But it's rather hard to change it for now. Furthermore, genetics alone does not fully explain IBD incidence. And that was quite nicely described in this paper by Manolio in 2009, where he described it as missing heritability of complex diseases. So what else is there? And that's where we come to the second layer of, let's say, the umbrella term would be still genetic material, which would be the transcriptome. Now, the transcriptome is essentially all the genetic material in the form of ribonucleic acid, RNA instead of DNA, that carry instructions for proteins. It's essentially the intermediate layer between the genome and the proteins being the actual effectors or the actual actors in well, what we consider the actors in diseases, for example. Each cell produces its own RNA, and as such, measuring all RNA gives insight into the behavior of a cell. But that has its, that incidentally also means that if you want to quantify the transcriptome as a way of for personalized medicine, you would need to know which tissue is important or which cell type is important. Each cell has its own RNA. If you were to look at the colon, or let's say the gastrointestinal tract, the transcriptome would be vastly different from what you would see in blood, and likely, in the case of IBD, would potentially be more telling in the gastrointestinal tract. But can you imagine that if you want to do diagnosis for IBD, you would need to constantly undergo a colonoscopy, which, practically speaking, is not, well, very feasible. Now, again, though, we have evidence from our colleagues in Leuven that transcriptomic data from colon tissue may predict response to vedolizumab prior to the start of treatment. So it does seem to suggest that it holds some predictive value. But what is the practical application thereof? That brings us to the epigenome. We have discussed the DNA at the bottom here and the transcriptome at the top here. Somehow they must interact with one another, and that is where the epigenome comes into play. Coined by Conrad Waddington around the 1930s, it was described as the developmental processes connecting the genotype with the phenotype. At a molecular level, they are essentially the modifications of the genome that affect the readability thereof. And essentially, it is therefore the interface between the genome and the transcriptome. It essentially sits in the middle here and directs which genes get expressed and which ones do not. And that makes it a very interesting an interesting modality to look at, because that's essentially what underlies the various cell types. Now, that brings us to the start of the METHYLOMIC project, the Omicron trial, because we had found that the DNA methylation of peripheral blood can predict vedolizumab and ustekinumab response prior to the start of treatment, which led to the setup of the METHYLOMIC project ultimately. Furthermore, what makes it even more interesting is that the whole concept of epigenetic is that it can be influenced by external factors, which could therefore suggest that you could maybe even change certain, or you could intervene at the epigenetic level, making well, either amending the loss of response or the phenotype of interest. Now, another modality that has been heavily researched within the field of IBD, the gut microbiome. It should come as no surprise that if you were to look at the gastrointestinal tract, you would want to look at what are the bacteria doing there. And IBD in and of itself has been associated with change in microbial diversity, indicated by several landmark papers showing that there is a well, as they call it, a very serious dysbiosis going on. Patients develop IBD. Now, akin to epigenetics, the plasticity of the microbiome remains an interesting avenue for therapy, and especially given that you can modulate the microbiome by changing one's diet, and perhaps in a more, let's say, clinical fashion, performing fecal microbial transplantation. We know, however, that if we look at fecal microbiome transplantations in ulcerative colitis, the effects of inducing remission remain modest. So whether it can be used for inducing remission still remains a question of interest. And this may also be related to the problem of that the fecal microbiome, the ones that you would see in the fecal sample, may be different from the mucosal microbiome. So those are things that we need to take into consideration when interrogating the gut microbiome. Now, how does this all relate to IBD? Well, as Virginia very nicely put it, IBD is likely a complex interplay between the aforementioned modalities, each with their own relationship. What I'm trying to say is that every patient is likely going to be different. It's going to be an interplay between genetics, where epigenetics, transcriptomics, and microbiome. This sounds, well, conceptually logical, but trying to put that into practice, trying to make sense of that remains challenging, both from a laboratory side, but also from a computational side. And especially from a financial side. Now, we live in a reasonably good time, given that there is at the same time also a huge emphasis on developing artificial intelligence and various computational models, which we heavily lean upon in trying to make sense of the relationships between all these modalities. METHYLOMIC being one of them, although METHYLOMIC does focus primarily on the epigenome. Now, throughout the presentation, I have already highlighted several challenges, but I feel that as an overarching concept, there are some challenges that need to be reiterated. Well, first of all, coming back to that each patient has its own disease, so to speak, reproducibility remains a challenge. Further, most of the studies are set up in a cross-sectional fashion. What this means is that we're just comparing cases and we're comparing controls. You only get therefore an association. We often don't know whether the observed difference that we find are causal, pathogenic. Just know that they are associated with the phenotype of interest, with the loss of response, or with the presence of IBD. Furthermore, we have a lot of findings that we identify at the lab, so to speak, but they don't often translate well into the clinic. And that in part may be related to the heterogeneity among patients. But it is also just vastly difficult to do these experiments because of the cost. Huge data sets are needed for the research. So ultimately, while we're trying to progress into personalized IBD treatment, understanding the complex interplay remains challenging for the aforementioned reasons. That doesn't mean that we are, I do not want to give a doomerist view on it all. We are proceeding onwards. We are moving forward. But we need to look at it very carefully. IBD indeed may need to be further characterized based on molecular signatures. Instead of considering as one disease, well, sure, you can consider that umbrella term. One may have to decide that there are various subsets of IBD. Similar developments have been occurring in the field of cancer, where, for instance, for AML, acute myeloid leukemia, they define P53 variants versus those that do not have that variant, or they have a methylation-induced colorectal cancer. Similar things I anticipate will happen in IBD at one stage as well. This will serve as a basis for subsequently tailoring the medication to IBD, because the reason why I believe, at least, why some patients do not respond is because they may present with a different term, with a different form of it. If you, for instance, have a variant in the JAK-STAT pathway, well, a JAK-STAT drug may work, but if the variant of interest that's causing your IBD may be elsewhere, well, then giving a JAK-STAT inhibitor would probably not be very beneficial. And finally, I would say that while targets or associations would be interesting, I feel that we would need to take this information, take it a step further. Can we use them as therapeutic targets, ultimately going the same path as the TNF pathway, the TL1A, and the IL-12/23 pathway? Now, with that being said, I would need to thank the following people for all their support throughout the previous years in setting up this project and for helping perform all the experiments. And I'd like to thank you for your attention.
Thank you, Dr. Andrew Lim, for this insightful presentation on the genomic biomarker for IBD. And now I give the floor to Roberto Saldana from IFCA, and he will speak on what matters to patients in all this discussion.
Yeah.
Roberto, the floor to you.
Thank you, Donella. Indeed, this science is too far away from the ordinary patients. However, and what we are doing is listening to our communities directly or with the support of our national members. When we listen to them, to the real patient, and we translated what they are saying and sharing with us, we realize that they are already speaking about how important is precision medicine, how we need to work on it, and also to spread the message and support every developer and the researchers to be successful in their works. Why precision medicine matters to patients? From the patient perspective, it's simple, because we need an accurate diagnosis. We need to accelerate the diagnosis, but not only to get the level of having IBD, because we also need to get something more from the clinical practice. We need to understand what kind of condition we have, what kind of phenotypes is going to show our conditions, what kind of symptoms we are going to experience, and also what kind of progression might we can have if we don't intercept the progression of the disease with a proper treatment. And this is, in fact, what is matters the most to patients, to be able to perceive the aggressivity of the condition, to understand that the diagnosis is real, that we didn't misunderstood the symptoms. Sometimes it takes time to get the diagnosis. People are going 2-3 different diagnoses before getting the IBD one, and this is hard, because in that time the damage is there, and if we don't intervene, maybe we are going to go through a surgery. We are going to go through unnecessary treatments and also unnecessary surgeries. Sometimes even permanent surgeries that is going to affect the whole life of the patients. But it's not always about us, the current patient. It's also about the future. We are concerned about what will happen with us, but also with our family members. What will happen with our children? Are they going to develop also the condition? Sometimes a patient not avoiding to have having children just because they are worried about that. If we can use precision medicine to stratify better our condition, and also the probability to predict those scenarios that are scaring us the most, maybe it would be ideal, because we are going to be adherent to the treatment, we are going to behave better, be more adherent, for example. And also, we are going to stop with the comparisons. Sometimes we are always asking, why me? Why are having those symptoms that are struggling us? However, some people are not dealing with those symptoms. Why? And those comparisons are also affecting our emotional status. Precision medicine is not for answering every question, but of course it's going to help to answer these emotional challenges that we are facing. About the, please, can you? Yeah, thank you. The other critical moment in the condition is also when we are monitoring and deciding the treatment we are going to use, because treatment selection without any other information is complicated. We don't know what will happen even when we are moving to the most newest treatments. We don't know if we are going to respond, if we are going to suffer side effects. We need to be sure that we are going to increase the efficacy of the treatment, the treatment we are using, and also at the same time, we need to be able to review the side effects. And because some side effects can be avoided thanks to this kind of predictions, thanks to this algorithm. And this is just what we are expecting from precision medicine, how we can improve the dose optimization, selecting the best frequency, the best doses for every treatment, for every patient, without using standard doses that maybe makes any sense for a specific patient. And the same with the patient that are not having the disease yet, the patient that are still coming to our community. We need to look for solutions that maybe help them to not get the disease, because we can stop the progression, because maybe in the future it's going to be possible. Or this is one of our most ambitious desire. Why not to speak about the curative instead of just stopping the flare and reduce and minimizing the symptoms. Precision medicine can also help us, and this is something that we expect a lot, to allocate the resources we have, economically speaking. Sometimes we are paying for external tests just for monitoring our condition, just because we don't know how we are progressing, or we don't have information in real time. When we are allocating properly our resources, maybe we can stop struggling economically speaking, because sometimes not all patients in every country have the same economic status that we have in Europe, for example. People are spending the money they don't have to monitor their condition, just for trying to find something that can help them to make better decisions, personal decisions, social, or any kind of decisions. And this is struggling emotionally the patients. Next, please. In their own patients' words, and this is exactly what I have been saying, people who have been suffering symptoms for many years, getting other diagnoses instead of the IBD, the proper one, just because it's share the same symptoms like diarrhea, bloody diarrhea, or even when sometimes we are saying in Europe that the diagnosis is not a problem anymore because we can get it in several months. That is not true if we don't take attention to the details. For example, women are experiencing a problem with the diagnosis. The time to get the diagnosis being a woman is the double, even more than when you are a man. And just because the clinicians are just saying that maybe they are experiencing this because they have psychological issues. This is real, this is happening, and this is part of the explanation why it is harder to get the diagnosis being a woman. Or when we have the side effect is not the same to be a teenager with an IBD than being an adult. When you need to face the side effect of the steroids being a teenager, being how your the body is changing when you are hanging out with your peers, it's more difficult. That is going to impact your condition, but also the rest of your life, because you are starting to learn how to live and socialize. If we can avoid that, and this is what patients are saying, it will be very helpful. At the end, what people want is to want stability, to reduce the flares, to reduce the symptoms, to get permanent remissions if we can remove completely the condition, and of course to have the ability to plan your life without any interruptions, just because you depend on a condition that you can control. Next, please. So, to the question why this matters to patients, is because it can offer us a better control, increase our quality of life without being afraid of what will happen in the next few days or even in the next years, to be able to planify and also to control our whole life. For families, is to understand how we are going to prevent with this information any event that we don't expect and want. And for the rest of the community, how we can also offer in a sustainable model for these patients that are still suffering, or for those ones who are still to come. So precision medicine empowers us to move beyond trial and error, that is the current strategy in clinical practice, towards a future of certainty and cure. And this is what is the most expected outcome that we can get if we are successful working on precision medicine in IBD. Thank you.
Thank you, Roberto, for this very insightful presentation and positive, I could say, presentation on the patient's expectations and what is in it for the patients. Seems as all is positive. So, I mean, I think in the discussion we can have a more thorough understanding of what the problems for the patients could be in personalized medicine. Then now I give the floor to Deborah Montague, and so Deborah is a lung cancer patient, I could say lung cancer survivor. She's the president of Lung Cancer Europe, and she's bringing to the discussion a lived experience from the oncology field. As you might know, personalized medicine is already very well advanced in cancer care, and still there are lots of burdens, obstacles to a full implementation of personalized medicine. So now I give the floor to Deborah. Deborah will present us her lived experience with cancer and with the diagnosis of cancer, and with the personalized medicine who helped her to survive a cancer. So Deborah, the word to you. Deborah?
Oh, apologies, I'm talking to myself. I put myself on mute. Apologies. Thank you very much indeed, Antonella, for the introduction and to the actual invitation to attend this evening's event. Can I get the next slide, please? Lovely. So I was asked to share my experience with my diagnosis, and as Antonella said, I am a lung cancer survivor. So this is my journey on 3 targeted treatments that are currently available for, well, actually the first one is no longer routinely prescribed for my particular lung cancer. It's gone out of fashion because there's been such a boom in treatment options being made available from once the first one came on board. It seemed to be the catalyst for many others. And so when we look at the landscape, it has changed enormously over the past 9 years. And I suppose that's one of the messages I would give, that when this, when you start go into this field and treatments start being available, maybe targeted treatments, then it's amazing how quickly the environment does change, and new treatments come along behind the originator. Next slide, please. So these are some of the side effects that these brilliant drugs give you. So I call it the gift that keeps on giving since my diagnosis. So you can see here that, so whilst they are superb treatment options, and for me, it made a world of difference. When I was first diagnosed, I envisaged being on chemo, and therefore sitting in a hospital chair once every 3 weeks and looking at the weekly availability or whether I would feel well enough to go out and socialize. So I actually bought myself a diary after my diagnosis, which was in the October for the following year. And I literally, while I was waiting for some test results, I literally mapped out week 1, week 2, week 3, and put on week 3 ready to socialize, because I'd heard such terrible things about chemo. Your first week, you're really sick. Second week was a little bit dodgy, but third week after treatment, you should be fine to socialize. And so that's how I mapped out my life going forward. And I then got the results of these tests I was waiting for, and I could have saved myself the price of a diary, because I was made aware of the targeted treatments, and I was put on my first one, crizotinib, and it changed my whole outlook in terms of being able to live with the disease for as long as possible. I was still at that time, because the crizotinib, the first one, was the only one available, and I was still told, well, you might have 3 years. So I was incredibly grateful, because otherwise the prognosis with chemo was maybe a year, maybe 12 months, maybe 18 months. So, and as you can see here, it's now over 9 years. So with the advent of targeted treatments, it has meant that I've got at least another 6 years and a couple of months of life that I wouldn't have had without a targeted treatment. And I think so, certainly in the field of lung cancer, it has transformed overall survivals and also progression-free survivals. Next slide, please. So for me, I think what it gives you is very much looking at, by targeting the treatments, you do see an enhanced effectiveness, and also patient responses do vary significantly. We know that some patients whiz through the treatment options that we currently have available. Others, they're on their first treatment. You saw there, alectinib, 8 years down the line. And I think that's one of the challenges with these personalized treatments, is we still don't know and can't predict accurately 9 years after the first one was made available, why some people do well and others don't. And there's still a lot to unbundle in these targeted treatments. But for some people, we've got patients now who've been on a single treatment for 11 years, which is incredible. And that, as I say, it's transformed the future. So the other interesting point is, I know I put up the side effects there on the first slide, but generally they're manageable and you can live with them and you can live well, far easier than the side effects of chemotherapy. So you do get a better quality of life on these targeted treatments. Next slide, please. So for me, yeah, I just think in terms of they improve overall survival. Definitely, we are living longer than certainly for the patients who are on the targeted treatments than we were previously. So that has already been well documented. So we know, a 12 to 18 month survival for a lot of patients on targeted treatments now is unheard of. You are looking at 7 years plus now. Next slide, please. So then I wanted to talk about some of that's the great news. They're great treatments when they're available, and with testing available to make sure that patients are identified on them. What are some of the limitations? I think you've got I will not be able to say this word at this time of the evening. Heterogeneity can be a challenge. We know, and it's interesting because some of these patients, when they have the tumor sample taken and the results come through, the challenge is that you've only taken that sample from a tiny area of that tumor, and so the rest of the tumor could have a very different makeup, and so it still leads and lends itself to an unpredictability in response, and that's partly we see that in people's varying response rates, definitely, and it does lead to treatment resistance. We know that none of these treatments will last forever, and so that is another challenge for the patients on these treatments. And yes, it's already been said earlier by, I think it was Professor Yin, said these treatments are incredibly expensive, and they are, and so there is the early days of targeted treatments for certain lung cancers, there was a reticence from payers to make them widely available, and that is definitely a challenge going forward. If hopefully you will have targeted treatments going for at some point in the future. Next slide, please. Yeah, in terms of, so I was asked to look at, well, what are the transferable lessons? And I think it's key to ensure that there is, and it's already been discussed around the importance of, precise subtyping, so we know which, especially in the area of IBD, which patients are appropriate and which aren't. I think one of the challenges, as I already alluded to, with payers is obviously if we need to set the scene accurately as to who and the size of the pool of patients who would likely to be eligible and respond to any targeted treatments that come forward in the future in your area. I think one of the other challenges is the availability of testing. If we look at the example of in the early days of biomarker testing for lung cancer, some of the pharmaceutical companies were providing that testing, those testing kits for free, in order to ensure that patients got access to the testing. So there's a chicken and egg. You need both the biomarker tests given approval, but also the treatments given approval. And I know from, if you look at the history of oncogene and say ALK positive and EGFR, I know that talking to pharma companies, there was a challenge in getting payers to understand the real benefits, the understanding of the quality of life improvements with patients, that they could go back to work and contribute to society in a much better way than they were able to if they were on chemotherapy. Next slide, please. So I think for me, when I did a little bit of work on this and tried to understand your environment, because I come from a position of lung cancer, and that's my area of expertise. IBD isn't, in terms of looking at it. I think, predictive biomarkers can help identify, treatment options. It seems to be that they're improving, which is fantastic news. And I'm delighted that there's another cohort of patients that might benefit from targeted treatments. That would be great. And the whole point of this for me is around avoiding ineffective treatments, and giving patients the best quality of life, really. That I think that's what we all want, really, and improve treatment success. The reality is, the more we can get the targeting accurate, and we're going to improve overall success rates with that. But that it's also making sure that it is widely available for all patient types. I live in the UK, and we have biomarker testing widely available, and yet we still know that patients in lower socioeconomic backgrounds have a much lower chance of actually receiving a biomarker test, and that is terrible. And we've had biomarker testing available for over 10 years now. So how can that be? So I would say those are definitely one of the lessons, is making sure that the messages are received by clinicians and payers very much further upstream than you envisage them needing to hear those messages, will lay the groundwork to make entry for any of these treatments going forward much easier, and hopefully more uniform than has happened previously. Next slide, please. Yep. And I was again looking at the technology advances that have taken place, looking at again, we're now starting to combine the whole genomic and all the data that's already been discussed, and Professor talked about that earlier on. So I'm not going to cover this. I'm going to move on quickly. Move on, please. There we are. Barriers, I've already talked about them already throughout, the cost challenges, the data integration issues, that sort of thing, and also clinician training, so that they understand what these treatments are and how they work and why they benefit patients. That's a whole ball game in itself in order to be able to achieve that. Next slide, please. But I think for me, if it was any take-home messages, start everything much earlier than you thought than anyone thinks you need to. Start the education about these potential treatments much further upstream with clinicians, payers, and patient populations as well, so that they're all on board when hopefully these new opportunities arise. So thank you.
Thank you, Deborah. Now, it was very interesting to see the evolution of your patient journey and the importance of precision medicine for you as a patient and for you as a patient advocate, because you combine the two roles. So now we were supposed to have a break, as we have we're running a bit late. I suggest to skip the break unless there is the majority of you. Maybe you can put it in the chat that you absolutely want a break. Okay, I think so. This is a positive sign that there is interest in the in the topic. So now I give the floor to Candida Cruz, who will make a contribution into the interactive discussion. And Candida Cruz is from IFCCA, the International Federation on Crohn and Colitis Associations. So, Candida, the floor to you. Candida?
I'm sorry, Candida, muted.
So Candida can, because we also don't see, at least I don't see her.
No sound. I was without, yeah.
Yes.
As I was telling, good afternoon. Thank you for introducing me. For us, I think this is a truly interesting topic because, but I think that this implementation will face at least three major challenges. Limiting access. First of all, each person is a unique individual, so there aren't enough validated biomarkers to know which treatment works for which patient. One is good for me, it can be not so good to other patient. Second, the existing tests are often not approved or reimbursed. Patient samples and data are underused, even though they could support the researchers. And this challenge, I think, matters a lot. Without reliable biomarkers, treatment remains trial and error instead of timely personalized care. Without real-world patient data, researchers cannot validate new diagnostics or drive meaningful breakthroughs. Without removing barriers to appropriate treatment, patients lose time and face avoidable risks and worse outcomes. The precision medicine in IBD means a lot for patients, I think, because faster and more accurate diagnosis will allow us to receive appropriate treatment sooner. Fewer trial and error treatments will result in less frustration, fewer side effects, and faster relief. More predictable disease control is improving also quality of life and fewer flare-ups. Fairer access to advanced therapies across all countries, allowing equal opportunities for the best care wherever patients live, and this is very important. On the other hand, removing legal and payer hurdles will allow biomarkers and precision medicine reach patients faster, reducing delays in effective treatment. With the precision medicine, people with IBD will indeed have a much better quality of life and hopefully what we want, the cure. Thank you.
Thank you, Candida. I mean, this is a very good introduction from the IBD patient perspective on the benefit of the direct benefit that personalized medicine would have on IBD patients. So now we have this session for the discussion, for the interactive discussion. I encourage you to, everybody in the audience to put a comment or questions in the chat or raise your hand if you would like to ask a direct question. And I'm not sure because I replied to some of the questions in the chat, if you can see my replies. While we do that, I can break the ice with some questions to the panelists. So I have a question for Andrew. I will start with Andrew, and then maybe the other panelists can jump in. So when researchers discover a potential biomarker, what are the steps needed from the biologic discovery, or for the original discovery, until this biomarker can be beneficial to patients in the clinic?
Okay, well, thank you for the question. Yeah, that's a very good question. I mean, I have always it's also something that I see a lot in the Department of Human Genetics. Essentially, it comes down to how is that study set up? What was the sample size? Because usually these studies are set up with a rather exploratory population size of maybe 10 on 10, 2020. So the initial plan would be to first extend the sample size and ideally do this across multiple centers, see whether your observation remains reproducible. I mean, it would be fun. It would be great if we find, let's say, a biomarker in Amsterdam, but it would still be terrible if that would not validate in, say, the UK or in Italy. Once that has proven successful, usually, and now I'm actually going more into what we do with the METHYLOMIC trial then, / the Omicron trial, where we would then try and see if the biomarker, say, can predict response or not. You would want to see, does it perform better than standard of care? Can you actually guide therapy? I think Virginia showed that very nicely with the AI-driven approach compared to the standard of care, where you set your threshold. Say, if your patient stays on medication for longer than this amount of time in say, the AI-driven approach compared to, say, standard of care, then it's superior. At that point in time, I think you have a relatively good scientific basis that what you have found is valid. But essentially, it boils down to you just want to see whether your observation generalizes to a larger population.
So that is, I mean, anyone else would like to add anything to this aspect? Because I mean, this is really because it's still Andrew, the development phase that you are talking about.
Yeah.
Because because in order to make a treatment, to make a personalized treatment really accessible to the patients, there are so many other steps. And for Deborah, and also my background is in oncology, we know well that unfortunately there is a huge gap between the advancement in science and what is then made available on the market for the patients. And in fact, another question for you would be, why do we often hear about promising new tools, but in practice we don't see them used? At least not as often as we would like to see them.
Yeah, no, I think that second, I'll first answer your second question, and then I'll loop back onto the first one. Why we don't often see it in, well, let's say the promise of certain biomarkers into the clinic. I suppose that one of the reasons could be that it is indeed not validated. But say that it is validated. Say that it went through, say, the trial of higher, and it's proven itself in a larger population. There is also the aspect of, and that's going back to your first question, getting this onto the clinic. I mean, if we go and find a biomarker and it's something that only one lab or very few labs in the world or in a particular country can do, it becomes logistically very challenging. And that's what we also see with the METHYLOMIC trial. I mean, these DNA methylation assays, it's not something that a routine diagnostic laboratory can do. You really need to run this at specialized centers. So what we're trying to do is we're trying to see whether we can distill this signal and convert it into a different test. Let me take the COVID example. I mean, we knew, for example, that the ACE2 receptor was actually a very good biomarker for whether someone did or did not have COVID. But that was initially discovered using sequencing-based approaches. Sequencing in and of itself is challenging, so they wanted to reduce the price and make it more available by converting it to the PCR test. Now, for gene expression, or the presence of such markers, that makes sense. For DNA methylation, those steps become a bit more involved. As an example, for DNA methylation, usually the process is you get your blood DNA, you need to perform a so-called bisulfite conversion, which already takes a day, and then you need to send it in for, say, the genetic analysis. And if you do that based on sequencing, that could take up to a week. So those things all factor into making it a viable product on the market. And that's also what makes it so challenging, I would say, to get from the idea that it is a very good biomarker up to something that we can actually use in clinic.
Yeah, thank you, Andrew. That's very clear. Virginia, now over to you. So what would a clinically useful biomarker look like to you in practical terms?
All right. Well, I believe that a meaningful biomarker should not just measure inflammation, but it should reveal the pathways driving it and indicate the therapy that most likely work in that patient. And clinically, this should be translated also in a simple and low-cost test that can provide actionable guidance before the treatment begins. So it should be reliable and responsive to treatment as a biomarker, and it should also be low-cost test.
Okay, so and also, so this is very interesting because then I would say really a long way to go still for IBD. Like for the METHYLOMIC project, considering, I mean, given it that the project will succeed in all the anticipated research questions, if it is 100% successful, after the end of the project, how long do you think we could see the impact on the patients? I mean, don't have the crystal ball, but at least try to anticipate.
So this is a great question. For clinical practice implementations, we have learned from other fields that there are prototypes. So if a trial is successful, the translation will depend on validation and regulatory approval and then accessibility. So realistically, it will a real clinical impact is likely to be seen in 10 years. Or approximately 10 years. This is what we have seen in successful experiences. Obviously, I don't have the crystal ball, but there are some kind of prototypes that we can replicate from what we have seen in other fields and from what we see also in drug development, not only biomarker development.
So thank you, Virginia. I think it was very important to set a sort of timeline to give patient organizations an horizon toward which work and prepare the community to accept and to use personalized medicine. So that was very, very, very good. Thank you. And now for a question for Roberto. So is there any way patient organizations could help at this stage science, so research at the like preclinical research and then clinical research?
Yeah, the the best support we can provide is validating the idea, validating the necessity to keep developing it, because if not, we are maybe in huge trouble. At the end, if something needs to arrive to the European market, for example, we need to prove how good it will be to solve real needs. And patients are the ones who can provide that feedback. Not the only ones, but maybe one of the most important stakeholders. So providing in which scenarios we can take advantage of this kind of science of tools, and also providing the outcomes we can expect, so that we can see the added therapeutic value that it provide us. That is one of the way we can support this development, and there are a lot of.
Yeah, so thank you. So that is from the research perspective, and that is very much the case. It is very important for research to be patient-centric. This is what you're saying, in order to be fully accepted and acceptable and fully feasible for the patients. So this is very important. Then when it comes to the implementation, sorry, I think somebody has the microphone on and I can hear. So when it comes to the implementation, then do you anticipate your role? Because I mean, you also as a patient group, it's important for IFCCA to start preparing the community, the IBD community, to accept personalized medicine, to accept or to push for it, to advocate for it. So where do you see a role for IBD?
Of course. First, we need to support the development, and also to go through the assessment of the value, so that the national government in any country can decide to reimburse the prices. Then, of course, we need to spread the message of how we can use this technology in our own benefits. Because if not, maybe we can make the same mistake that we made in the past, like the ones we are already seeing with the fecal calprotectin test. There are patients that have not used them right now in many years. The physicians that are not using them because I don't know why. Maybe because of the price in some health systems, maybe because they are not aware of the benefits, or because any other reason. So we need to learn from other experiences to avoid the same mistakes, and maybe anticipating, spreading the message with the real cases, practical cases. Maybe we can accelerate the implementation and the real use of this technology.
Yeah. And in fact, so this is very, very true, Roberto. In fact, I can bring you an example that is very indicative, and it's really a learning example for us in the oncology sector, but you could also use it in the IBD. So what we did, there was a survey focusing on, it was a survey on genomic testing and breast cancer. We had chosen breast cancer because it's the most common, where women are more aware, normally are more health confident, and so on. And we focused on 5 countries: Germany, Italy, France, Spain, and the UK. So 5 big, important, easy countries. And we did this genomic test. We did this survey on genomic test and breast cancer. We had about 1,500 respondents, and it came out that 4 out of 5 women with breast cancer eligible from that genomic test, 4 out of 5 were not told by their oncologist the genomic test was an option in countries where the test was available and reimbursed. So there is really a lot
of example of what we shouldn't do.
Yes.
Yeah. That is what I am afraid of, and then we need to anticipate that scenario to change the pathway and go through another scenario, the best one for us. Because if not, maybe the technology is going to be there, but we are not going to use it, and it's going to be a pity.
And so we need health literacy, we need shared decision making. I don't know if you, Deborah or Roberto, from the patient perspective, if you have any examples of health literacy programs or shared decision making activities that you have been implementing.
I would like to recommend and raise awareness of an FT3 resource from Testing to Targeted Treatments organization, that are a global organization, and they have a patient information booklet on biomarker testing and its value, and they make it freely available for patient organizations to translate, to one, adapt to their particular needs. You can take as much of the information or as little as you wish, and turn the resource into your own, and put your own logo on as an organization. And also they will translate it into as many languages as you need. So that's a really good starting point in terms of that patient education piece. So raising awareness and raising expectation for patients as well. But yeah, it never ceases to amaze me that we can still find clinicians who aren't offering patients biomarker testing that is routinely available in the UK. It's shameful, really. And as I said when I was presenting, the earlier you start that education piece with clinicians and payers, then it will pay off. And I think one of the things that, as one of the resources that has been useful for the payers, is where you map out a potential patient journey early on, and that is shared across multiple payers across a geography, so that they can clearly see what needs to be in place and where, rather than everybody working from a blank piece of paper. I would say, take, as soon as you've got examples, share them widely, so that it saves some of the pain. I'd like to say it saves all of it. Unfortunately, it won't, but it can. And take the learns from oncology. They're at the forefront, and the early adopters of biomarker testing. If you need patient advocates, maybe look for some within the oncology world to meet with key decision makers, be that payers, clinicians, or whoever you're trying to convince. Then they would be the best people to really extol the benefits of biomarker testing and then getting onto the right treatment.
Thank you, Deborah. Yeah,
an extra 6 years so far.
Yeah, thanks, Deborah. And Roberto, so from your side, what is the importance of shared decision making when discussing treatment options with the gastroenterologist?
Information. I'm really convinced that shared decision making is important. However, we need to be aware that there is an important part of the information that we really need to make the decision that is lacking. For example, And this is changing, but it's still happening. What is the best drug for reducing pain in IBD? What is the best drug for reducing incontinence? If we don't have access to that information, what kind of shared decision making we can make? So we need to still keep generating information regarding to what is mattering the most from the patient perspective. Because if not, maybe we can encourage people to communicate with the physician, to convince the physician to be open. However, what kind of discussion they are going to have without the information, practical information? That is the real challenge, and we are still dealing with that gap between what we really need in clinical practice and what the discussion we can have with all information we are gathering from clinical trial or even with the observational studies. At least we know that IBD clinics that are using questionnaires to collect PROs are improving the communication with the patients. Patients who are used to report PROs, they communicate better with the patient, that they are focusing on what is most important, they share more valuable information with the physicians. However, that is not common, and it's complicated. Shared decision making is not really possible right now, and it's not because there is a real option or that armamentarium with different treatment. It's because there is missing information there.
Yeah, thank you, Roberto. And that is true. And the information is health literacy, comes with health literacy, because if the doctors share information with the patients, then it's important for the patients to understand what the doctors are talking about. So now we are approaching the end of the session. Before closing, I would ask each panelist to share in 30 seconds what would be what they would need to enhance personalized medicine from each one perspective. 30 seconds to the optimal level. So like if you wanted to get personalized medicine to the top, what would you need? What, from your perspective, is needed? Who wants to start? Well, Virginia, you start.
All right. What we need for in the implementation of precision medicine is this the question.
From your perspective,
so, from my perspective as a clinician, I think we need resources to build large international longitudinal cohort and publicly available data set where scientists can work on. And we need, of course, but this is something that we are trying to do, the involvement of patients from the very early steps of protocols, trial implementation before the real practice application of what we need.
Thank you.
I believe that's. Thank you.
Andrew.
I think for me as a basic translational scientist, it would be getting a cohort of large number of patients that provide, say, the various omics data that we have at hand. Say that within the context of our research questions. So say that we are looking into that response, and it would be great that we have a large cohort where we have genetics, material, epigenetics, transcriptomics, etc. I mean, it falls within the adage of the more data, the better. It sounds very corny. But if we do want to make inferences and subclassifications of patients, I feel that we must start with a large data set at first, whereafter we can distill the information necessary.
Thank you, Andrew. Deborah, 2 words.
Patient involvement.
Excellent. Good. Roberto, and to you the last word from IFCA in the IBD community.
Okay, I will say data and of course users. If you don't have the user and a purpose for them to support them, it's not going to be useful.
Excellent. And with this, so data is summarizing what Virginia and Andrew also mentioned, and users, so patients willing and available to use personalized medicine. So I thank you, all the participants, the organizers. There is another Antonella in the back office, and Maria Stella, and all the speakers, of course. Thank you very much for the clarity of the exposition and for really making to the point of the session. So thank you, everybody.
Thank you.
Thank you.
Thank you.